So the first thing that we're going to look at two different types of cellular division. So we have mitosis and meiosis. Mitosis is also known as somatic cell division. It's basically cell replacement, and this happens everywhere except in your testes if you're a man and in ovaries if you're a woman. So what we're doing is we're taking a diploid cell, diploid meaning 2N. So what this N is, this is the number of chromosomes that we have in our body. We have 23 pairs of chromosomes. So if we're looking at chromosome number 6, we have a chromosome 6 from mom and a chromosome 6 from dad. So if we take all the different chromosomes, which we have chromosome 1, 2, 3, 4, 5, 6, 7, all the way through 23, and we have a copy each from mom and dad, that's what gives us 46 chromosomes total with 23 homologous pairs, we call them. Homologous meaning homologs, meaning similar in structures. So all you need to know, this term diploid, it comes from us having 2 copies of each chromosome. We have 23 chromosomes with a copy of each, so we get 46 chromosomes. So these are our diploid cells. Meiosis, you start with a diploid cell, 46 chromosomes, 2 pairs of each of the 23 copies, and you're going to end up with 4 haploid, meaning you only have 23 chromosomes in each cell. You don't have 23 pairs, you have 23 chromosomes. Why is that important? Because if I have a sperm with 23 chromosomes and I have an egg with 23 chromosomes, when these blend back, then I once again have 23 pairs. So if this is 2 times n being 23, then I get back to my original 46 chromosomes. So if you're a human, it means that you're diploid and you have 2n chromosomes in all of your cells except your sperm and egg cells. So if all of you needs to have 2n, then whenever you're being made as a baby, when you mix the sperm and when you mix the egg, the sperm needs to have 23 and the egg needs to have 23 so that you only have 46. You don't want multiple chromosomes. If you guys have ever heard of trisomy 21 or Down syndrome, this is from an extra copy of chromosome 21 being put into a cell. Okay. This is my example for mitosis. So what I'm trying to get at here is we have copies. Remember, I said that we have 23 pairs, and each of our chromosomes has a copy from mom and a copy from dad. So what I did is I just said, let's look at chromosome number 3 in general. So chromosome number 3, here I have a copy of mom's and a copy of dad's. We know that as we do cellular division, we are going to replicate our DNA, so let's show that. This is before we divide the cell, so I've replicated my DNA. And now when these separate, I still have a copy of number 3 from mom and a copy of number 3 from dad, and a copy of number 3 from mom and a copy of number 3 for dad. So this is how we're getting haploid cells. We start with diploid cells. We start with 1 diploid cell, and then we go to 2 diploid cells. So we don't lose any genetic material. That's important because if you're a skin cell, you don't want to lose all your genetic material because it has information that you're coding for skin stuff. So this is how we conserve genetic material in all our cells. We copy it, boom, there's your copy, we split it into 2 cells. Again, this will copy, split it into 2 cells. Copy, split it into 2 cells. In meiosis, that's not how it works. So instead, let's just say, for example, we're looking at chromosome number 3. So copy from mom, copy from dad. They replicate, okay, so they replicate and they replicate. But now when they're pulled into each cell, you can see one of the cells has 2 copies of mom's and one of the cells has 2 copies of dad's. So we've gone from 2n, because remember we have 23 chromosomes, to now just being n, because we have 23 chromosomes each with a copy of itself. So this is where it gets confusing for people because you don't see this difference unless you've taken a genetics class where in mitosis you're going to get equal division because they all line up on top of each other. But here they line up, the chromosomes line up next to each other, the pairs line up next to each other. So in this first cellular division, you're already getting a reduction in your genetic material. We don't divide again. We're not going to we don't replicate our DNA again. We are about to divide, but we don't replicate our DNA again. So we don't get extra copies like, if I was going to go here, we don't get extra copies like this. So since we don't do that and we just split this up, then you say, well, how are these different then? Because we say that meiosis gives us 4 genetically different gametes. But this looks the same to me because this is 3 and this is 3. Well, that's where if you've taken a genetics class, you've heard of this term crossing over. So in crossing over, you'll have your chromosomes line up just like they did here. And part of this chromosome 3 and part of this chromosome 3 will swap. So what ends up happening is you have chromosome 3 from your mom, and then you have chromosome 3 from your mom with a little bit of chromosome 3 from your dad. Same over here. Chromosome 3 from your dad, chromosome 3 from your dad with a little bit of chromosome 3 from your mom. So this is what's giving us our genetic diversity. This is not the same as this, which is not the same as this, which is not the same as this. And again, you could write every number in here. 1, 2, 3, 4, all through 23. I hope this helps, this division of genetic information. So let's do a quick recap with some words now that we have words and pictures. So mitosis. Mitosis takes us from a diploid cell. We replicate our DNA and we divide, and then we have 2 diploid cells. Now, we start with a diploid cell in meiosis. This is meiosis right here. Diploid, we replicate our DNA, but we have this weird lining up of the chromosomes, which means we're going to have a genetic reduction material. So we have 2 haploids. These haploids aren't going to replicate their DNA, they're just going to divide, and this is where we get 4 haploid cells. I hope this helps. And now we're moving into cellular division, which is this Premed. Whether it's whether it's mitosis or meiosis, regardless, we're going to follow Premed, which stands for prophase, metaphase, anaphase, and telophase. If we're doing meiosis, then we have a prophase 1, a metaphase 1, anaphase 1, telophase 1. And then instead of replicating your DNA again, you're just going to go straight back into prophase. This will be prophase 2, metaphase 2, anaphase 2, telophase 2. Regardless whether it's mitosis or meiosis, let's talk about what happens during prophase versus metaphase versus anaphase, etc. Prophase. So prophase, this is saying chromosome. So the chromosome condenses into what you think looks like a chromosome. Normally, chromosomes are just in this loose form called chromatin like this. And it's just all over the place. And all of that will condense into something that looks like this that you're used to. All right. So prophase, always think chromosomes are condensing into what we know as like these structures of chromosomes. So chromosome condenses from chromatin to a chromosome, and we also get the formation of the mitotic spindle. So the mitotic spindle, if we come here and we draw a cell. In the video we talked about a centriole, so the centriole, you should go back to the organelles video if you need help with that. This is our nucleus. In prophase, our nuclear membrane is going to divide, so we have all this nuclear envelope is going to disappear, nuclear membrane is going to disappear, and then we're just going to get this lining up of chromosomes. So here's chromosome 1, here's chromosome 2, chromosome 3. The centrioles of the mitotic spindle, the mitotic spindle comes from the centrioles. They're going to reach to the middle of these chromosomes and pull them that way, and then we have another one here to pull this way. Okay? That's what your centrioles are doing. So they haven't pulled anything apart yet. We're just really in this prophase form. You don't have the lining up of the chromosomes here, so you might have a chromosome there, maybe one here, over there, there. But in prophase, you start this mitotic spindle, which just means these fibers are starting to extend. If we move into metaphase, I like to think of as middle phase because the chromosomes are going to line up on the equatorial plate, which is just the middle of the cell. So chromosomes meet or line up in the equatorial plate. So they would meet in the middle. So let's modify this picture. The mitotic spindle is going to keep extending through metaphase as our chromosomes start to line up in the middle. And right now we're looking at mitosis versus meiosis, and I'll modify this picture for meiosis after. All right, so we have our prophase. Nuclear membrane disappears, chromatin condenses into chromosomes, mitotic spindle begins to form. Metaphase, lining up, continuation of the extension of the mitotic spindle. Next is anaphase. Anaphase are where the chromosomes are going to get pulled apart to opposite sides. And in this case, these are actually, so if I'm looking at this, I'll say this is chromosome 1 from mom, and this is chromosome 1 from dad. So each of these chromosomes have been replicated to have two sister chromatids. So if I look at, this is chromosome 1 from mom, this is chromosome 1 from dad, they replicate. So now it's still chromosome 1 from mom and still chromosome 1 from dad, but they have two sister chromatids. So this is a sister chromatid with this, and this is a sister chromatid with this. So sister chromatids are pulled apart during anaphase for mitosis. So C-somes, or you could say sister chromatids pulled apart. And then that telophase is just where you have that cytokinesis event. So in telophase, we'll say that a membrane is going to form around each set of DNA. So they've lined up in the middle. So our cell, we had prophase, nuclear membrane disappeared, chromosomes start getting like this, nice and condensed, mitotic spindle starts forming. Metaphase, everything lines up, boom, 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 mitotic spindle keeps going. Anaphase, we just showed. Now we have all these single chromatids on this side and this side. And now for telophase, we can put a nuclear membrane around it, or a nuclear envelope. So there's one and there's one. And then we get the cytokinesis action. Boom, boom. And now we have two nuclei all conserving our genetic material. Boom, 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 boom. How is meiosis different? So this was our prophase and metaphase sort of example, and I show you here, if I say that this is chromosome 1 from mom, this is chromosome 1 from dad, we're always going to get a chromosome 1 from mom and a chromosome 1 from dad, chromosome 1 from mom, chromosome 1 from dad in this split. So you never lose genetic material. Now instead, if we're looking at meiosis, alright, if you stuck with me through that last video, we talked about this idea of crossing over. So instead of all of our chromosomes lining up like this, now we're going to have chromosome 1 from mom and chromosome 1 from dad lining up next to each other. So these are homologous chromosomes lining up next to each other. This is 1 and this is 1 versus this, we saw this is 1 and this is 1. We can change that chromosome number so it doesn't get confusing. And we can say this is chromosome number 3 from dad, and this is chromosome number 3 from mom. Okay, so this is for mitosis. Now that we're in meiosis, let's change these from 1 to a 3. So this is chromosome 3 from mom, and this is chromosome 3 from dad. Remember, that allowed us to do this crossing over thing that's going to give us a genetic diversity, and that's happening between prophase 1 and metaphase 1. So let's compare the pictures as we pull things apart. So here we had sister chromatids being apart. This is mitosis. In meiosis, we have homologous chromosomes being pulled apart. So these homologous chromosomes have crossed over, they have some switching, so we have genetic diversity. But now when they get pulled apart in anaphase, we have mom's copy of number 3 going to one cell and dad's copy of number 3 going to the other cell. So you immediately get a reduction of genetic material because you don't have both mom's 3 and dad's 3 in the same cell. So we get through our telophase 1 and we'd have 2 haploid cells. So this would have mom's 3 and this would have dad's 3. But don't forget, they've been copied already during that meiosis 1. So they have copies of themselves. And then we don't replicate DNA, we just go straight into our division, and now we have 4 cells. This has moms, this has, and remember we'd get a little bit of blue on this because of the crossing over. And we'd have fully blue here, and then this would have a little bit of red to show crossing over. Now we've made 4 haploid, genetically unique gametes.